Welcome to Solihull Sixth Form College Open Evening and the Biology Department. Today we're going to take you through A-level biology with us at Solihull Sixth Form College. I'll give you an overview of the course and we'll show you some of the practical work that we do in the college. Okay, I'm going to start a presentation now to take you through the things that we do in the college. Okay, so this session is going to take approximately 30 minutes. The aim is to give you an idea of what it would be like studying the course here at Solihull. And if you have any questions at all, type them into the Q&A box and one of my colleagues will either attempt to answer it or they'll pass it to me at the end of our session and I'll go through some of the commoner questions with some answers at the end. Okay, so the A-level course is run by the exam board AQA, which many of you will be doing your GCSEs with. We just offer the A-level course, so there's no opportunity to do the AS course. The A-level course is designed to continue your studies from GCSE, either combined science or separate sciences. So you certainly shouldn't worry if you're only studying the combined science at the moment. There's no bar to you doing A-level biology. The course is broken up into 2 years. The year 12, you'll start off by looking at biological molecules. Then we'll study cells, particularly focusing on the cells of the immune system. Then we look at exchange of substances with their environment, starting with cell transport and moving on to exchange, gas exchange in a variety of organisms and transport in organisms. And finally, in year 12, we finish off by starting a view of genetic information, variation, and relationships between organisms. In year 13, we broaden our look at energy transfer between organisms, looking at respiration and photosynthesis in some detail, as well as energy transfers in ecosystems. Then we move on to looking at the response of organisms to changes in the environment, both the internal and the external environment, looking at the nervous system and how homeostasis works. We'll build on what we did in year 12 and then look at some more genetics and evolution and how that affects populations, particularly in ecosystems. And finally, at the end of year 13, we'll look at the control of gene expression and really focus on gene technology, which is a big area of research in biology at the moment. The course is assessed by 100% examination, which is taken at the end of year 13. You'll have 3 exam papers covering different topics, with paper 1 covering the year 12 content, paper 2 covering the year 13 content, and then paper 3 having an overview over both years and looking at how the different topics interact with each other. Also, during the 2 years, you'll be assessed on your practical skills. And at the end of that, you'll receive a certificate of practical competence if you pass that part of the course. Which brings me on to the practical work that you'll be studying during the course. We do a range of practicals, including the required practicals. I'm going to look now at some of the practical activities with various members of the department. And we're going to start with looking at plasmolysis of red onion cells with Dr. Richard Orton. So I'll let Richard go through that practical with you.
My name is Richard. I'm one of the biology teachers at the Sixth Form College, and I'm going to demonstrate a practical that we do, which will be to use a red onion, which we have here, and we're going to look at plasmolysis in red onion cells. Is to make a slide using red onion. The thing about the red onion is you see it's red, red colour because of the pigment inside the cells, and that will enable us to see what's happening in under the microscope quite easily. Onions is that you can peel them and get thin strips of tissue very easily, as you can see. So that is one cell thick. So when you put that under the microscope, you just get a single cell, layer of cells, which is very easy to see what's going on inside them. And I've got one there. Put that onto the microscope and lay it down. Try and get it as flat as possible. It's actually probably a bit too big for me. It doesn't come off so nicely. We'll use it. Then what we're going to do is add a few drops of molar sodium chloride, so that salt solution which is well concentrated. And to protect everything, we'll put a cover slip on top. So we'll lower that down gently. That will flatten everything and spread the solution around, and that then will be prepared. Okay, so this slide shows the appearance of red onion cells under a light microscope at medium power with normal conditions for it. And if you have a look at the cells, you can see the cell wall quite clearly outlined here. And inside the cell, you can see that the cytoplasm, which contains the pigment called betalain, which is giving it its pink colour, is pushed more or less completely against the cell wall. So these cells would be described as being turgid, and this is the normal state of a plant cell when it's got plenty of water, and it enables the cells to have a strong structure and provide stability, etc. for the plant. And the question is, what happens when you put this normal tissue into a solution that contains a concentrated solution of salt? And in the next slide, you'll see what happens after it's been 1 molar salt solution has been added. And you can see it's quite a clear difference. So you still got some cells which are more or less as they were previously, but most of the cells, you can see they changed quite dramatically. So there's a lot more space inside the cells, and the cytoplasm is much more concentrated. You can see this much more clearly if we go up to the high power. So this is times 400. Okay, so here's the same tissue under the high power, and you can see the differences between different sorts of cells quite clearly. This cell here is being pretty much unaffected by the salt solution. So you can see I'm outlining roughly where the cell wall would be, and you can see that the cytoplasm in there is being pushed still up against the cell wall. So there's no space inside that cell at all. Now if you compare this cell, which is still pretty turgid, with this one here, I'll outline the cell wall again, you can see quite a big difference. So here you've got quite a lot of space between them, and if I use this, you can see here the outline of the cell surface membrane within this cell, and it's been pulled away from the cell wall. So there is quite a large space in this sort of area here where the cytoplasm was and is no more. And the other thing to think about is that as the cytoplasm has shrunk, the volume has got less, the amount of pigment inside that cell is still retained the same. So it's got darker, it's become more concentrated. So you can recognize the plasmalized cells very easily because they're much darker in pigmentation than the other cells would be. It's also quite interesting to think about what would be in that space where the cross is, where the cytoplasm used to be. It's not air, it's not a vacuum, it's actually filled with solution. And some of that solution will be the salt solution that we put on there, but quite a lot of it is going to be the water which has come out of the cytoplasm by osmosis, crossed that cell surface membrane, and is now filling that space. And that's the reason that the cytoplasm has shrunk. It's because it's lost water. As the water has moved out by osmosis, it has shrunk back, and the space is then filled with that water. So this is a slide showing you plasmalized tissue. next question to think about is what would happen if we flushed out the salt solution and replaced it with deionized water, which is effectively distilled water. And that is what you can see in the next slide, which will be a video of that actual experiment. Right, so you can see we just flushed the cells with distilled water, and you can see that the volume of the cytoplasm, the dark splodges, is starting to increase inside each of the cells, and eventually you'll see it pushing against the cell walls. So there we go, so most of them are getting completely full now. And you can see that the volume of the cytoplasm, the dark splodges, is starting to increase inside each of the cells, and eventually you'll see it pushing against the cell walls. So there we go, so most of them are getting completely full now, and that's more or less it. Okay, so what have we just seen happening? I'm hoping that you would suggest that it has something to do with osmosis. And GCSE, you probably think of osmosis as being the movement of water molecules from an area of high concentration to low concentration across a semi-permeable membrane. That is a normal definition that you would use, but there are problems with that definition. For example, what does semi-permeable mean? Does that mean half the membrane is permeable, or just a bit of it, or what? And concentration is usually measured by what's dissolved in the water rather than the water itself. So when you come on to A level, you need a better definition, and this is the one that we'd expect you to use. So at A level, osmosis is the movement of water from a high water potential to a low water potential across a selectively permeable membrane. So how does that relate to what you have just been looking at? Well, this is a plant cell, and this is going to represent the onion cells that you've just been looking at. The solution which has the highest possible water potential would be deionized water, and that is what we have just been bathing the cells in. And deionized water has the highest possible water potential because it's got nothing dissolved in it. Basically, it is water and nothing else. By contrast, the cytoplasm will have a much lower water potential, and that is because it's going to contain amino acids, sugars, proteins, soluble proteins, ions, and all sorts of things, all of which will increase the concentration of the solutes in the cytoplasm and therefore lower the water potential of the cytoplasm. And as a consequence of that, what we'd expect to see happening is that water will cross that selectively permeable cell surface membrane by osmosis and start to move into the cytoplasm. So you can see here the water is moving into the cytoplasm, and that is going to increase the volume of the cytoplasm. So what you then see happening is that the volume of the cytoplasm will start to increase, and the cell surface membrane will therefore expand and start to push against the walls of the cell. And so we end up back where we were started at the beginning of the experiment, with the cells being turgid, as they're full of water at that point there. Okay, so that is the explanation of what you have just seen. Thank you for listening, and I hope you enjoy the rest of the session.
I'm Claire Drake and I teach biology at the Sixth Form College. We're going to have a look at a practical to do I'm Claire Drake and I teach biology at the Sixth Form College. We're going to have a look at a practical to do with the effect of temperature on the permeability of membranes using beetroot. At GCSE, you will have learnt about cell membranes and their role in controlling which substances are able to enter and leave a cell. At A level, we build on this understanding by studying the components of membranes and how the properties of the components affect the membrane's function. This is a detailed drawing of a model of the cell membrane called the fluid mosaic model. It shows that the membrane is made up of many phospholipid molecules arranged in two layers with proteins embedded in it. When looking at the diagram from either side of the membrane, it looks like the components are arranged into a mosaic pattern. Individual components are able to slide past each other within the same layer of the membrane. This is why we describe the membrane as fluid. This is a beetroot, and the reason we're going to use beetroot for this experiment is that it's got a very strong pigment that is kept within the cells, and normally it remains within the cells because the membrane is not permeable to this pigment. So what we're going to do, we're going to cut some cores of beetroot to use for the experiment. But in order to make sure that we're running a valid experiment, we need to make sure that these cores are the same size as each other to control this variable. So I'm going to use a cork borer, and this is going to take cores out of beetroot for me. Okay. Right, and then I need to remove the core from the cork borer. Okay, so one nice long continuous core there, and then I'm going to carefully measure centimetres of beetroot core. Right, I'm going to take off the end because that's got a bit of the skin of the beetroot on, and that would affect my results. I'm going to use the ruler to carefully measure a centimetre at a time. And now I've got my pieces of beetroot which have got an equal surface area to each other. The next problem you can see is that it's already staining my fingers. So when I've cut the beetroot core, it's actually released some of the pigment from the cells that I've broken as I did that. So I'm going to wash the cores in this beaker of distilled water to make sure that any of the pigment that is on the outside of these cores is no longer there before I start the experiment. So these are the 3 cores of beetroot that I've cut that have been washed and blotted, and now they've been placed into 10 millilitres of water. It's important that the volume of the water is the same in each of these tubes. These will now be placed into water baths and left for 5 minutes, at which point we will tip the liquid out of these tubes into new ones, and then we will have a look at the colour and the density of the pigment in these tubes. So while we're waiting for those tubes to incubate in the water baths, we'll have a quick look at what we would expect to happen in this experiment in general, with 2 similar cores that have been put into. The one on the right hand side of the screen as you look at it is in cold water that's come straight from the tap, and the one that's on the left hand side of the screen is in water that has come out of a boiling kettle. So this is a time-lapse video of what's going on, and what you see is that there is a faster rate of release of pigment from the core that is in the hotter water than the core that is in the colder water. At the end we give them a quick swirl to distribute the pigment within the test tubes, so you can see the difference in concentration of pigment between the 2 tubes. These are the tubes that we've incubated with the cores in water baths at different temperatures where we knew what the temperature was. So the tube that is on the left hand side of the screen as you look at it, the palest tube is from a water bath that was at 22 degrees, the one in the centre is from a water bath that was at 43 degrees, and the one on the right hand side of the screen as you look at it is from a water bath that was at 62 degrees. And we can clearly see that there is a difference between the tubes, and that we go from lightest, so least concentrated, in the tube on the left hand side of the screen up to most concentrated or darkest on the right hand side of the screen, where we know the water bath was hotter. But this is a qualitative judgment. I can't tell you by eye how much more pigment has been released in the second tube compared to the first tube, or the third tube compared to the second and first tube. These are the tubes that you just saw. And we're going to use a colorimeter to get quantitative data from our experiment. The thing we need to do is to calibrate the colorimeter. Now what happens with this is a light is transmitted through the sample, and how much pigment there is in the sample, more or less light will be absorbed. However, the gloss of the tubes will also absorb some of the light, so we need the machine to take away the absorbance of the tube and the water itself from the absorbance of the whole sample containment, so that we know how much of that absorbance is due to pigment alone. So first off, I'm going to put this tube, which just contains distilled water, into the colorimeter, and I'm going to press reference, and that tells the colorimeter that that's the amount of absorbance you'd expect just for water and glass alone. So next up, I'm going to put in the tube of the liquid that's come out of the water bath at 22 degrees centigrade. And I'm going to pop that into the colorimeter, and I'm going to press T for test. And the colorimeter tells me that the absorbance for that sample due to the pigment is 0.05. And this is in arbitrary units. So if I take that one out of the colorimeter, the next one I'm going to test is this tube here, which came from the 43 degree. Right, you can see, but it would be difficult to put a number on it. Right. So I'm going to sample the colorimeter and press test again. Right. And we can see that it's 0.1 arbitrary units. So in actuality, this is pigment in this tube compared to one in the water bath at 22 degrees C. And finally, I'm going to test the tube that came out of the water bath at 62 degrees centigrade. Pop that in the colorimeter. And the absorbance for that tube is 1 arbitrary unit. So it's a much, much stronger concentration of pigment within that tube than in either of the other tubes that we tested.
So what do these results show us is happening? So at the top of this slide, you can see the results from our experiment plotted onto a graph, and beneath it you can see the results from somebody else doing a very similar experiment, but they've done more temperatures than I have just now. Right. And you can see a pattern in this graph. From about 0 degrees up to 30 degrees, maybe up to 40 degrees, you've got a very slight increase in the amount of pigment leaking from the beetroot cores. However, after 40 degrees, you find a quicker release of pigment from the beetroot cells. So what is it that the temperature is doing which causes this difference? From GCSE, you already know that increasing temperature increases the kinetic energy of particles. So if we start to heat the membrane up at these lower temperatures, that increased heat energy is transferred to increased kinetic energy, and you get faster movement of the particles in the lipid bilayer past each other. And very occasionally, that means that small cells. So what is it that the temperature is doing which causes this difference? From GCSE, you already know that increasing temperature.
So, coming back to how you will study biology with us at Solihull Sixth Form College. Each week you'll have 3 1.5 hour lessons plus a 30-minute support session in a week. So that's 5 hours of directed study in college per week for your A-level study. We have a team, a number of people who are all trained biology specialists teaching on the course, so you'll have an expert to teach you. Many of the teachers are also examiners for AQA, so we have a wealth of experience with answering and carrying out analysis of the exam papers. You'll be taught in specific science laboratories, so all the lessons take place in a laboratory. We have plenty of lab equipment, so you'll get plenty of hands-on practical experience when you're at the college. We use a wide range of teaching resources, from textbooks through to online resources, and you'll have access to those resources. Throughout the 2 years, we do carry out key assessments to make sure you're on track and you're working at your best level of understanding. There will be 5 in the first year, and then there will be another 4 key assessments in the second year. So you can see here a range of examples of some of the resources. Some of these are online, such as Caboodle, Moodle, Exam Pro. Some of these are textbooks, the AQA textbooks, and Biological Science Review, which is available online and also in the Learning Resource Centre. Just takes you a bit further in your studies. You'll also get booklets of exam questions and guidance throughout the course. We do require certain course qualifications for you to study biology with us. You'll require 2 GCSEs at 6 or above, plus 3 other GCSEs at 4 or above, and a 4 in English language GCSE. To study biology, you will also require a 6 in maths and a 6 in biology and either chemistry or physics, or if you're studying combined science, a 6/6 or above. So, in summary, biology is the study of all life, so we don't just focus on human biology. We look at everything from biochemistry to the evolution of life over the history of the Earth. It's an excellent foundation for progression onto many different medical and caring professions, from medicine, veterinary science, pharmacy, dentistry, as well as those such as nursing, physiotherapy, and other caring professions. There are also a number of other biotechnology fields to go into and environmental fields that all feed from biology. There are a number of transferable skills that you get from studying biology, such as curiosity, problem solving, critical analysis. It helps with improving your math skills, literacy, and ICT skills are all things that you need to study biology effectively, and you will need in employment. Biology complements all A-level combinations from maths, chemistry and physics to psychology, PE and art. We also offer a range of enrichment links. These help boost your progression chances at university or apprenticeships, as employers value the skills you develop by doing something extracurricular. In biology, we also encourage students to study core maths if they are not doing A-level maths, EPQ, where a number of students will do a biology-related EPQ. We also run a pre-medical school for those wanting to apply for medicine, and we have a STEM club, and we have entries into the Biology Olympiad. So you have a range of opportunities for you at Solihull. There's a number of careers and progression that you can see on the screen that biology leads to, and we have had students entering many of these professions and a number of destinations of universities where our students have gone to. And we've got a few minutes left, so just to finish off, I'm going to ask if there are any other questions. If you want to type them in to the Q&A, I can see if I can answer any of them now. Just trying. Let's have a look. I think my colleague has managed to answer many of your questions as you've gone along. So I'd like to thank you all for attending. I noticed some of you may have had some problems with the sound on some of the video clips. Hopefully we can upload this onto the college website at some point and you'll be able to access it again if you want to listen to the videos again. Thank you for attending the session. Good luck with your GCSEs, and good luck for your college applications.